IBM and the Mayo Clinic have brought state-of-the-art computer science, natural language processing, and language understanding principles to complex queries. This enables text analysis, indexing, and retrieval by keyword, phrase, or concept to the Mayo Clinic's 20 million electronic clinical notes, allowing researchers to electronically retrieve concepts and knowledge from these records without reading each one. Mayo Clinic's clinical notes have been normalized and annotated, and the annotations found are matched to MESH concept IDs. MESH is a standardized, controlled vocabulary thesaurus created by the National Library of Medicine. Beyond a free text search, concept searches include synonyms, narrower terms, broader terms, and related terms. Dr. Sanders wants to search the clinical notes that discuss the patient's AML diagnosis and have a mention of prednisone. He will do a MESH concept ID search of the clinical notes for AML. Note the capabilities to expand both the context attributes and ontology hierarchy. Dr. Sanders also checks the clinical notes for a mention of prednisone in the text by including a free text search. The term replacement feature corrects the spelling. Synonym expansion function is also available. However, Dr. Sanders chooses not to expand his search beyond prednisone. He runs his query and finds 4 patients that match his criteria with multiple clinical notes. The notes can be viewed from the results screen in the document viewer. Dr. Sanders can easily see the topics that he searched for, with annotations highlighted in yellow and free text highlighted in blue. He can also easily see additional annotations by selecting the highlighting for these. Note the annotation for chest pain in the lower right-hand corner, indicating a certainty value of -1. This confirms the patient did not exhibit this symptom as shown in the clinical document. This is an example of the strengths of concept search over free text search. Dr. Sanders saves this query so that he can access it later. Next, he repeats the process to identify the control group of patients that have been treated for infectious mononucleosis. For this demonstration, we'll open a query that contains conditions for both the experimental and the control group. Dr. Sanders uses the group OR function, combining these conditions, to find patients that have either AML or infectious mononucleosis. The results of this query show 4 patients found for each group. Of course, these results are an inadequate subject number for any meaningful research, but are sufficient for the purposes of this demonstration. Combining genotype and phenotype data into one system will allow physicians and researchers to draw inferences previously undetectable. Dr. Sanders determines whether microarray data exists for the patients identified in both groups. He also adds a check that the bioassay data meets an initial quality control condition of the RNA material as measured by the absorbance ratio of the tissue used. For this, he requests an absorbance 260/280 ratio between 1.9 and 2.0. In addition, he requests information he would like to see when the query returns its matches. These include the experiment name, the disease under investigation, the name of the principal investigator, additional quality control parameters, and the chip type used in the microarray experiment. Having defined all the conditions of interest, Dr. Sanders runs the query. The results show microarray data for the 8 unique patients identified earlier, 4 with AML and 4 with the benign condition, infectious mononucleosis. Dr. Sanders is satisfied with the experimental and control patient cohorts identified by both phenotype and genotype data. He now wishes to perform various analyses of the bioassay data for these patients to look for gene expression differences between his experimental and control groups. First, he applies an experimental quality control analysis which utilizes SAS software. He determines that these quality controls meet his criteria, so he moves on to additional analyses of the microarray data. To further analyze this data, he performs a single-factor analysis, where the experimental factor field will be the ICD-9 code. This routine is a long-running analysis that can take some time to complete, so he schedules it to run in the background. The single-factor analysis, a Mayo Clinic-developed routine, performs a statistical comparison between 2 groups, for example, AML versus control, by implementing a permutation test. After the analysis routine completes, Dr. Sanders goes to My Query Results, where the results have been stored. In this table, all of the more than 12,000 genes on the microarray are summarized. He can easily take these results and visualize them in various ways. He selects the single-factor charts routine and runs this with a specific request to return the top 10 genes showing the greatest fold change between disease and normal state. In the first figure, the so-called volcano plot, he can see that a number of genes show marked differences in the log change expression, either downregulation, shown on the left, or upregulation, shown to the right of the 0 on the x-axis. For many of these genes, the down or upregulation is persistent among all patients, resulting in a significant p-value, as shown on the y-axis. Although not shown, additional charts can be viewed, such as confidence interval plots and chromosome location plots. In the next analysis, Dr. Sanders views the same volcano plot using Spotfire. By clicking Open, he can send the table of data he's analyzing directly into the Spotfire application. Spotfire allows him to drill down from all genes into a single specific gene of interest by zooming into the plot and selecting an individual gene. He selects a gene markedly downregulated in AML versus the control group. He reviews the specific information regarding this gene as shown on the right. Dr. Sanders identifies other genes of interest that show marked differences between his experimental and control groups, closes Spotfire, and returns to the Data Discovery and Query Builder software. Dr. Sanders has identified genes with differences in gene expression in patients with malignant and benign hematological conditions. Now he decides to search PubMed for articles that reference these genes. He chooses the rows of interest and selects the gene literature search among the analysis routines. He initially searches for only one of the genes of interest. This literature search, based on gene name, is implemented by PubGene. The results of this gene literature search show summaries for each article, and on this page, Dr. Sanders has the option of selecting an article to review the abstract in more detail. As he was interested in AML, a hematological malignancy, the first article, by Hill and colleagues, immediately attracts his attention. By clicking on it, he can read that his gene of interest has been studied in 3 family members. Each had a common chromosomal defect and developed a hematological malignancy. These results confirm that further investigation into these genes as targets for new diagnostic or therapeutics for AML is justified. Here again, a conclusion reached in minutes versus weeks or months. This concludes the demonstration of new biomedical informatics tools, developed by IBM in collaboration with the Mayo Clinic. We have shown how these tools allow querying of data, such as laboratory results, clinical notes, and demographic, diagnostic, and genomic information. We have seen how results can be analyzed using homegrown software, or how they can be routed into commonly used analysis packages. This solution leverages key components of the IBM Healthcare and Life Sciences Clinical Genomics offering, which is built on proven technologies, including IBM DB2 Universal Database, WebSphere Application Server, a web-enabled interface called Data Discovery and Query Builder, and IBM eServer and storage technology. These new biomedical informatics tools allow analysis of data across a wide spectrum, from bench to bedside. And the result, a future where medical practice is improved by knowledge generated from the integration of diverse clinical and biomedical data, leading to more effective and targeted treatments, and ultimately, personalized medicine.